Alright, hi everyone. This is the third video lecture for cell signaling part 1. We are in the middle of discussing this question here, which is what are the particular types of signaling molecules that our cells and bodies produce, and in addition, how do these signaling molecules actually go about mediating changes in cell behavior? In the last video, we talked about this first general class of signaling molecules, that is, molecules that are capable of crossing directly through the plasma membrane, and we talked about how many of them, specifically hormones, function by binding and activating intracellular receptors that are called nuclear receptors, which in turn leads to changes in gene expression and in turn changes in cell behavior. Now what we want to do is start talking about this second general class of signaling molecules, that is, signaling molecules that are not capable of crossing through the plasma membrane because they are polar. And so here we're going to talk about what these molecules are and fundamentally how they modulate changes in cell behavior. To get us started, I'll tell you that these molecules represent the widest variety of cell signaling molecules that are produced in animals. And they include, for example, some hormones, which is to say hormones that are not capable of passing through the plasma membrane. They include neurotransmitters, and they include a large group of signaling molecules that are called peptide growth factors. What I want to do here is introduce you to what I mean by peptide growth factors, because it turns out that these things are super important in regulating cell behavior, so I just want to be clear on what these things are. To do so, I'm going to show you an example of a peptide growth factor, which is epidermal growth factor, or EGF. This is a peptide growth factor that we know plays very important roles in regulating animal cell proliferation, both during embryonic development, as well as in adult organisms. This over here is a depiction of EGF, and hopefully it's obvious that we call these things peptide growth factors because they are indeed polypeptides. They're usually fairly small, so EGF is only about 53 amino acids in length. And indeed, the way that our cells produce these things is they have genes for the various peptide growth factors. So there's an EGF gene that cells will transcribe and translate to generate EGF protein. And then that EGF protein will be secreted so that it can go and modulate the behavior of another target cell. So that's fundamentally how the various peptide growth factors, what they're made up of and where they come from. If we now start thinking about, well, how do all of these molecules regulate cell behavior, we can start thinking about it by pointing out that the mechanism through which these molecules work is fundamentally the same, or at least very similar. What they're all going to do fundamentally is they're going to bind to the extracellular domain of some cell surface receptor, and the key is that is in turn going to trigger a cascade of reactions inside the cell, and that cascade of reactions is what's going to drive the change in cell behavior. With that said, I want to come back to this image that we looked at before when we first started thinking about cell signaling in concept, and point out that these images actually included depictions of cell surface receptors. And so these squiggly lines here are supposed to be the cell surface receptors, and as you can see, they're supposed to represent transmembrane proteins that have an extracellular domain that binds to an extracellular signaling molecule. And so that's sort of the first step in an extracellular signaling molecule initiating a change in cell behavior by this mechanism we're talking about. At the same time, note that these cell surface receptors have an intracellular domain. And the key is when that molecule binds to the extracellular domain of the receptor, that's going to trigger some cascade of reactions within the cell through this intracellular domain. This image is not depicting those reactions, but now what we're going to do is begin thinking about what those reactions would be that are initiated by this intracellular domain of the receptor. To think about this, I'm going to start with this image here, which sort of is going to give us a broad overview of the types of things that the intracellular domain of a cell surface receptor can do to drive changes in gene expression. So you have to, or to drive changes in cell behavior. So you have to imagine that this is supposed to be a cell, of course, and this is supposed to be a cell surface receptor, which again, transmembrane protein with an extracellular domain that binds to a particular extracellular signaling molecule, which would be called the receptor's ligand. The intracellular domain has all of these lines coming off of it, and what that's supposed to be representing is the fact that when the signaling molecule binds to the receptor, that's going to initiate some type of activity in the intracellular domain of the receptor, which in turn is going to trigger this cascade of reactions. The key is that those cascades of reactions can sort of do one of two general things inside the cell. On the one hand, it can lead to activation of what's called an intracellular signaling pathway, which in turn leads to a change in the function of pre-existing proteins inside the cell. And so if you start changing the function or activity of pre-existing proteins, then that of course can alter the cytoplasmic machinery inside the cell, which in turn can alter the cell's behavior. The other thing that the cell surface receptors can trigger is, again, through intracellular signaling pathways, they can drive changes in gene expression. And so that's what this is intended to indicate. And of course, if you start driving changes in gene expression, that can lead to an alteration in the proteins that are present inside the cell, which obviously can also alter the cytoplasmic machinery and in turn drive changes in cell behavior. So these are sort of the two broad ways in which cell surface receptors can drive changes in cell behavior upon binding to an extracellular signaling molecule. Again, either through modulating the activity of pre-existing proteins or through driving changes in gene expression. With that in mind, that raises basically the last two questions we're going to think about for cell signaling. This first question is going to be the focus of the remainder of cell signaling part 1. Which is we're going to think in a little bit more detail about what exactly these cell surface receptors are and the way in which they work to initiate these intracellular signaling pathways. After that, during cell signaling part 2, we are going to look in more detail at what these intracellular signaling pathways are exactly and how they are activated by these receptors and then drive these changes that we just went through. Okay, so to finish off this video lecture for this particular video lecture, we're going to start getting into defining what those cell surface receptors are, and then we're going to talk about one class of those receptors. So to think generally about these receptors, let's start by saying that mammalian genomes actually encode thousands of different cell surface receptors. So we have many, many different cell surface receptors, and we categorize these things based on their mechanism of activation. And so, for example, there are about 1,000 cell surface receptors in cells that are classified as G-protein coupled receptors, or GPCRs. And these things serve as receptors for all kinds of things, from growth factors to neurotransmitters to hormones and other extracellular signaling molecules. So they all have different ligands, but they are all considered GPCRs because they share a common mechanism of activation. Likewise, there's another group of cell surface receptors that are called receptor protein tyrosine kinases, or RTKs. So there are several of these. They all differ in the particular ligand that they bind to. However, they share a common mechanism of activation. Beyond these two groups, there are others as well, and so I'm just pointing that out here. We're not going to get into these others because we just don't have the time to. So instead what we're going to do is we're going to focus on the GPCRs and the RTKs to try and understand how these things work. So what I'm going to do with the rest of this video is focus on the GPCRs and hopefully help you understand what they are and how they work. So as I just mentioned, mammalian genomes encode approximately 1,000 different GPCRs, each of them which binds to a different ligand, which is to say a different extracellular signaling molecule. However, despite the fact that they bind different ligands, they all share a common structure and mechanism of activation. This image here shows the common structure that you'll find in all of these 1,000 GPCRs. As you can see, it's a transmembrane protein, of course, but it consists of 7 transmembrane domains. It has an extracellular domain, which, as we said, is going to contain the ligand binding site, which is to say the site that binds to its particular extracellular signaling molecule. And then in addition, of course, it's going to have a cytoplasmic domain or a cytosolic domain, which is responsible for initiating signaling when the receptor is bound to a ligand. The way that these receptors work is it turns out their activation requires the activation of the receptor requires the activation of a group of proteins that we call G-proteins. And G-proteins is really just another term that is often used to refer to GTP binding proteins. If we take a closer look at the G-proteins that work with these GPCRs, we can note that they are heterotrimeric protein complexes that consist of 3 subunits that are called the alpha subunit, the beta subunit, and the gamma subunit. And so shown here is a depiction of the G-proteins that you will find associated with the GPCRs. To give you a sense of the G-protein complexity, just note that our genomes can produce, or they encode, 21 different alpha subunits. So we have 21 different alpha subunit genes, 6 different beta subunit genes, and 12 different gamma subunit genes. And so the key here is that these different alpha, beta, and gamma subunits can come together in different combinations, in turn generating an enormous complexity in the G protein complexes that can be formed. Since they are GTP binding proteins, G protein activity is of course regulated by allosteric binding to GTP and GDP, as we've talked about many times in the course so far. So again, when G proteins are bound to GDP, they are generally inactive. However, when they're bound to GTP, that is when they are in their active state. The key here in linking up these G proteins to the GPCR is it turns out that the inactive GDP-bound form of G proteins are found associated with and regulated by GPCRs. And so this is the state that you would find a G protein within a cell when the cell is not in the presence of a extracellular signaling molecule for this particular receptor. So they are stuck there. What happens here is it turns out that the GPCRs, their intracellular domain exhibits GEF activity, which remember the GTP exchange factor activity, which is responsible for activating G proteins. And the way this works is that GPCRs will catalyze GTP exchange on the G proteins only when a ligand is bound to it, and that in turn results in G protein activation. So what happens is when a ligand or growth factor for GPCR is present, it binds to the extracellular domain of the GPCR. That induces a conformational change which activates its GEF activity in its intracellular domain, and that in turn activates the G protein. Right? So it initiates GTP-GDP exchange, and that in turn puts the G protein into its active state. This image here is from your textbook, which is just another depiction of what I just showed you. This is supposed to represent the GPCR, and it does not have its ligand present, and therefore it is associated with a G protein on the intracellular domain that is bound to GDP. Like we said, ligand binding, however, is going to stimulate the GEF activity of the receptor, which remember the intracellular domain here is what has the GEF activity. And remember, GEF stands for GTP exchange factor. So shown here is what happens when the ligand for the GPCR is present. The GEF activity is activated, which in turn puts the G proteins into a GTP-bound state. The key is, when the G proteins are active, one of the things they do is they dissociate from the GPCR, and so they are released, and indeed the subunits fall apart like this is shown. So the alpha subunit is released, and the beta and gamma subunit are released and stay in contact. When they're released, these things go on to regulate a wide variety of different targets, including enzymes and ion channels, which typically is mediated through protein-protein interactions. So essentially that's what happens when these things are activated. These G proteins go off and start regulating targets. However, this process can be reversed because it turns out that cells have a gap, which remember stands for GTPase activating protein, a gap called RGS. And what RGS does is it converts G proteins back into their inactive state by hydrolyzing that third phosphate on the alpha subunit of the G protein. And that in turn leads to the reassociation of the G proteins into their heterotrimeric complex and association with the receptor. If the receptor does not have a ligand present, then the G proteins will remain there. If the receptor does have a ligand present, well then it will reactivate the G proteins and send them off to regulate their targets again. Alright, so that is fundamentally the mechanism by which these GPCRs work. And so what we're going to do is from here, we're going to go on to talk about the second class of receptors, the RTKs. That will finish off cell signaling part 2, and then we will come back around to talk about signal transduction cascades that are activated by these receptors.